Okay, so welcome everyone to our webinar today on exosomes and polynucleotides, the cross-protocol breakthroughs that we have developed with Revive and X with your favorite polynucleotide. Okay. So I want to say hello to our head office team, our international partners that are joining us today. I can see them coming in one by one, and all our practitioners and academies throughout the UK and Ireland. Thank you for joining. We've got lots of new practitioners and members that have joined today. So a big welcome to you. I'd like to introduce myself. I'm Nita McEvoy. I am the official distributor for the Revive NX portfolio for Charm Meditech. We have 5 main modalities. Today we are going to go through exosomes. We don't have a polynucleotide. We haven't got that in our future plans at the moment, but we will show you how to cross protocol treatments and how to make it work and get the best treatment, maximizing your profit margins, your time, and giving your clients the best regenerative outcomes. So I'm Nita McEvoy. I am the lead trainer for UK and Ireland and the official distributor for the product portfolio. And I'm introduced today to you Dr. Keiron Lord, who is our medical expert and medical lead for UK and Ireland. We're honoured to have him on board. He'll be going through all the scientific parts of the webinar. We're doing a light recap on exosomes, and then we're going into a deep dive on polynucleotides and how to combine them in your treatment protocols and roadmaps for your clients. Dr. Lord will be covering all the science, and then I'll be taking over the last part, short part of the presentation for your business integration. Because let's face it, we're here to make money as well as give our clients the best treatments and stay at the forefront of the aesthetics industry. So I hope you find this insightful. We will be giving you a replay at the end once we've uploaded it onto YouTube. Now, this webinar is recorded, so if you're not comfortable with having your face on the cameras, please put your cameras off, and please stay mute throughout the webinar. Feel free to drop the questions in. We'll have an opportunity for question and answers with Dr. Lord towards the end. And I'll hand it over to you, Dr. Lord. Thank you for having us today.
All right, thank you for inviting me along again. So, hello everyone. I'm Keiron. I've been an aesthetics doctor for around 10 years, particularly focusing on regenerative aesthetics and longevity medicine, and implementing treatments such as exosomes and polynucleotides into my clinic. I have done for a few years now that really are kind of changing the way that our skin ages. We're kind of beyond that time now where it was just about filling faces and just adding volume and freezing with Botox. And people are coming in now to clinics wanting these regenerative treatments. They don't want to look like a different person. They don't want to look over volumized. The last thing they want is for someone to know they've had anything done. And so instead, they're coming in just saying, I want to look fresher. I want to look like I did 10 years ago. What can you do for me? And that's where things like exosomes and polynucleotides come in. The problem with that is that there are a lot of companies out there making polynucleotides and exosomes because they know people are asking for them. And you're often overwhelmed by these products on Instagram, on Facebook, TikTok, by pharmacies marketing and sending things to you. And you often have no idea what's a good one, what's not a good one, what's the protocol, can I use this with that? And so when we put these webinars together, it was basically to allow us to kind of cover some of that stuff that you might not necessarily know. Um, and with that in mind, this is all about sort of the science behind the treatments. So you understand them and you can explain them to your clients. But it also means that, yes, we're talking about Revive because I'm on here with Revive talking about the exosomes. That's because the Revive NX exosomes fits that category or those criteria that make it a good exosome. And this is why we can talk about polynucleotides today, because even though Revive don't do a polynucleotide, it's still important to know about. So I'm going to teach you a little bit about how you choose the right products and why, and how you can layer these products to get the best results for your clients. So let's see if I can make this move because it seems to just want to move go down. No, hang on. Slide control. Why won't it let me move to the next slide? Oh, hang on. There we go. So we often treat the symptoms of aging when we look at the skin. So when we're looking at skin, when people come into our clinics, we see fine lines and wrinkles, we see volume loss, we think about putting fillers in the cheeks, we think about putting Botox around the eyes. But that is just treating the symptoms. Really, we've got to think about the underlying cause. What's causing those symptoms? And if we treat that, we're going to get a much better result for our clients. Aging skin begins at a cellular level. It's not just about, oh, you're getting old and therefore you're getting some changes to your skin. It really is at the cellular level. So my background before I was a doctor was in biochemistry and biomedical science. So I have a real passion for this cellular biology and cellular aging processes. And I feel like if we can understand why our cells age, we can start to use treatments cleverly to intervene in these steps. So, in this talk, we'll cover how regenerative treatments like exosomes and polynucleotides are evolving and allowing us to change the way our cells age. If you look at your cellular structures, we have energy sources in the cells which produce energy for us to carry out our daily functions, as we'll see shortly. This starts to decline with age, which means our cells can't repair themselves and replicate themselves like they used to, which means damage is carried forward and is shown as aging. We have stresses like UV damage, which causes further DNA damage and pigmentation. We have the breakdown of collagen as a result of, often if it's female, it's often sort of estrogen loss around menopause time. And for the rest of us, it's external pollutants and environmental stressors like UV damage, which can break down the collagen. But by knowing this, we know we can then intervene and slow down the process. So it isn't just all about time. It's about the cell communication system. It's about the cells communicating with each other, breaking down. And when this signaling process slows down and is less effective than it used to be, we find that we get a lot of the signs of aging. So we have cells in our body called fibroblasts. These fibroblasts live in our skin, and fibroblasts are responsible for creating collagen and elastin, but mostly collagen. Fibroblasts start to decline from around the age of 25 years old. They don't go anywhere, they just go into a hibernation or a senescent stage. So they go to sleep. And so it means that when they're asleep, they're not functioning like they used to. They're not producing the collagen like they used to. And we get a declining collagen level in our skin. As you can see from those two pictures, on the left-hand side, you've got nice long strands of collagen that are keeping the skin supported. This acts like a scaffold to hold the skin up. On the right-hand side, you can see aging skin, where we've got small fragments of collagen that have been broken up over time. New collagen hasn't been replacing it. And so we've started to notice signs of aging, which is this sagging of the skin, fine lines and wrinkles, because you haven't got that structural element underneath anymore.
Sorry, Kieran, I think I
Yeah.
can't see your slideshow. I don't know if it's just me.
Oh,
It's kind of come off screen.
hmm. Let me try and go back. Let me see. Let me stop sharing and try and share again, maybe.
Am I the only one that's seeing this?
It might be this. I don't know. Let me try again.
Yep, you're
Can
back.
you see that?
Yep, you're back.
Yeah, just interrupt me if it goes off again. Uh, right. So yeah, you can see the collagen there. You can see the the breakup of the strands of collagen, which is then responsible for these the loss of structure and the wrinkling of the skin. When we lose collagen, when we lose the function of these fibroblasts, not only does collagen reduce, healing slows down, and we lose our skin's integrity. So why are we talking about this? Well, that's because if we understand this process, when we see fine lines and wrinkles, we're not thinking about, oh, let's add filler. By the way, I'm fine with filler. I think filler's still got a place in regenerative aesthetics. It's just got to be done cleverly. We're not jumping straight in with filler because think about it. What's filler doing in this process? Filler is just an inner product. It's sitting in the cracks and temporarily volumizing, but it's doing nothing to support the underlying skin. It's doing nothing to stimulate collagen production. It's doing nothing to re-energize the cells. So what I want you to do at the end of today's talk is to understand that actually these aging processes in the skin are happening, are responsible for the signs and symptoms that you see in clinic, and that actually there are treatments out there that target that problem. So we're not just treating the symptoms, we're treating the underlying cause. Um, Hopefully my slides have moved on. Can you see the next slide on there? Oh, you're muted.
Yes, I can still see it in
Yeah.
the
All
screen.
right. I'll assume you can see it unless you tell me otherwise now then. So we've covered cells such as fibroblasts in the skin, which are important for collagen. Next, we need to look at the role of mitochondria in the skin. Mitochondria are in every single cell of our body. They are the energy factories of the cells. Our cells need energy to carry out every single daily function. So that is often repair of DNA damage. Every single day, our cells are exposed to damage from environmental stresses like UV light. This causes mutations within the gene. If the genes in our cells are mutated and not repaired, this is carried forward and can result in um, cellular aging, cellular damage, cancers and things like that. And so one of the main responsibilities for our cells is to make sure we're correcting that damage each time, so we're not carrying forward DNA damage. This is highly, and it requires a high amount of energy to do this. So we need to be able to produce energy. And we do this with the mitochondria. The mitochondria produce ATP. This is dependent on a molecule called NAD+, which you may have heard of. Um, our NAD levels decline as we age, and our mitochondria become less efficient. As our mitochondria become less efficient, they produce less energy. And as this happens, our cells have to start to prioritize what they're going to do with the limited energy that they've got. So it's important to replicate because the cells have a finite lifespan and you can't just not have a cell to replace that skin cell. So it has to replicate, but it probably doesn't have the energy anymore to be quite as stringent when it comes to identifying DNA damage and repairing that damage. It also doesn't really have the energy to produce the extracellular matrix, so the collagen, the elastin that we used to have. It also maybe doesn't have the energy to make all the signaling molecules, the proteins we need, or the carbohydrates that go into our cell wall. So the cells replicate and we keep getting new skin cells, but these skin cells are carrying forward a lot of DNA damage and aren't working as efficiently as they once were. So how can our exosomes play a part here? Well, exosomes, especially those with any active growth factors, can help restore the mitochondrial performance. They can stimulate the mitochondria to work more efficiently. It's like giving your car a good service. It will work much better after the service, and that's what the exosomes can do. Um, so. Another thing we need to consider, so we've looked at the lack of or the reduction in collagen as a result of fibroblast inactivity. We've looked at the mitochondria aging. We've looked at the energy reduction in the cells and how that can impact our cellular health. The next thing we need to look at is this inflammatory loop. So inflammation is around, inflammation happens in our bodies, and this is one of the key driving factors for many disease processes, not just in our skin but everywhere. So, low-grade inflammation can be responsible for certain cancers, can be responsible for certain inflammatory conditions like endometriosis and bowel disease. It can be responsible for Alzheimer's disease. And this is an entirely huge talk all in itself, which we won't go into right now. But inflammation driven by insulin or high insulin is a massive risk factor, an evolving area now in research looking at this and how we can control that because it's been linked to a lot of disease processes. When we look at other things that contribute to aging, we have two pathways. One we can do something about, one we're kind of quite limited. And so if you look at these people over in a lot of them in the States who are very much longevity, biohacking experts and understand everything that they're doing, but they're spending an absolute fortune every single year on trying to prevent aging, trying to halt the aging process. And unfortunately, as things are right now, we're unable to do that. And that's because of some of these intrinsic factors. So it doesn't matter what you try and do, there's a certain amount of aging that will happen anyway. Okay. So intrinsic aging is things like telomere shortening. In our cells, we have our DNA, and at the end of the DNA, we have telomeres. That's extra bits of DNA that don't do anything. Doesn't matter whether we've got them or not. But every time we replicate our cells, our DNA shortens. So the telomeres are there so that the genes, or so that the DNA that is cleaved off and shortened is stuff that was redundant anyway and we didn't need. If we didn't have telomeres, as that shortened, we'd be losing genes that are important for coding for proteins and various things in our body. So telomere shortening is something that happens. There's not much we can do to stop that. We can slow it down with certain treatments, but we can't do anything to stop it completely. Oxidative stress, we can reduce, but we can't stop it. So oxidative stress comes from our environment. Hormonal decline is inevitable. We can give HRT, but it's never the same as your own endogenous hormones. So with hormone decline, that's men and women. Women go through the menopause. Women lose 1.5 to 2% of their collagen every single year from the age of 25. And once they hit menopause, in the first 5 years of menopause, they lose 30% of whatever's remaining. When men go through the andropause or the menopause or whatever you want to call it, men lose testosterone, and that can have different impacts, but can have similar results. And autophagy dysfunction is basically our body's way of getting rid of damaged and old cells. It's the recycling center. It gets rid of the old cells and makes new ones. And this becomes less efficient as we age. This results in fine lines, wrinkles, dryness, reduced elasticity, reduced structure. Now, those are the things we can't stop. These things are going to be happening anyway, okay? But what we can do is slow them down. And by impacting the extrinsic factors, the extrinsic aging processes, we can reduce the strain and therefore slow down the aging. So things like UV damage. How many of us have people who come in to clinic and have a treatment and then say, "How long till I can go on the sunbed?" You're having skin treatments to stop these signs of aging. Don't go on sunbeds. But avoiding UV exposure, even on sort of cold winter days, the sun's out, you will be exposed to UV rays. Make sure your clients are using a good SPF. Smoking, one of the most harmful things you can do for your body and for particularly for your skin. And so by trying to encourage people to reduce smoking, it's really, really helpful to help the skin, but also their overall health. Pollution, if we're living in cities, there's always high pollution. Even in some of the rural areas now, we see high levels of pollution. Um, and our diet plays a big part. And I think a diet is something we can definitely influence very easily. You know, smoking, often people find it difficult to quit. Pollution is in our environment, and short of moving house and living somewhere on a remote island off the coast of Scotland, you may struggle. But diet, we can certainly do something about. So, avoiding foods that spike your insulin. So, insulin, as I said before, is a massive driver for inflammation in the body. There's loads of studies about it. I could go on about it all day and I won't because it'll bore you to death. However, it's a really interesting topic if it's something that you want to read up about. High insulin levels is not normal. We have not evolved as a species to have high amounts of sugar in our diet. We've evolved to have small amounts of sugar very often, very frequent, infrequently when we find a bit of fruit somewhere when we're foraging. We get high sugar, our body has that sugar, it boosts the insulin so that we can get the sugar into ourselves and we can store it for starvation times. In the environment we live in now, we are exposed to sugar in our diets all the time. Therefore, we've always got raised insulin in our blood. Our body doesn't like having raised insulin. Insulin causes inflammation, and that inflammation causes a lot of chronic problems in our body, um, including things to the skin like pigmentation and collagen decline. So, addressing the diet by reducing those insulin spikes, so trying to have less processed foods, trying to avoid these refined sugars, is one way of doing it. Thinking about intermittent fasting, because that also reduces your insulin spikes, and making sensible food choices, including your fruit and vegetables and things like that. So we can do things like this to impact that sign of aging. The intrinsic aging is a little bit more tricky. So exosomes and how exosomes are going to play a part in this. Let me just get all the stuff on the slides. So um, this is where we move in aesthetics from simply plumping and hydrating the skin to genuinely activating cellular renewal and reducing the signs of aging at a cellular level. Exosomes work on a cell-to-cell signaling pathway. So they communicate to cells to repair, to regenerate, and to reset the skin. So essentially, think of them as cellular messengers. And I'm going to tell you a little bit about how they work in the next slide or two. So exosomes are lipid envelopes, and we have them in our body anyway. So when our cells want to communicate with each other, they send out exosomes. Our cell membrane is made of lipid. If we want to communicate to the cell next door to us, what we will do is our DNA creates a message in mRNA or some other form. It sends it to the outside, to the very edges of the cell. The cell buds off a little bit of lipid envelope as a little sphere. Inside that sphere is the mRNA molecules. That goes on to the next cell. The lipid envelope fuses to the cell membrane and releases that message inside the membrane. And then that cell interprets the message. This is really useful in biology for things like when we have infections. So if we get a sore throat in the winter and a cell is infected with a virus, it will create a message that says, there's a virus knocking about, I've been infected, but you guys who are all around me need to protect yourselves and make sure you're not infected. That message is transported in an exosome. It goes to the surrounding cells, and the signals in that exosome will tell the surrounding cells, okay, put up your barriers, switch off those proteins, increase that protein, harden up the exterior wall of the cell, that sort of message to say, don't get infected. And that's all an exosome is. And what clever people in the lab have done is have altered that message. So think of it as an envelope. We've intercepted that message. We've removed the message that's inside. We've replaced it with one of our own to tell the cells what we want it to do, improve your skin barrier, switch on fibroblasts, make more collagen, etc. And we've resealed the envelope and sent it on. And that's what an exosome is. Revive NX, the reason why I like Revive and the reason why I use it in my clinic is that there's a few things that make an exosome good. One is where it comes from. Two is the amount of exosomes in the actual vial that you're getting. And finally, unique to this is the nitrogen 1 technology. So when you're looking for an exosome product, look at the amount of exosome in there because it makes sense. The more exosomes you've got in a vial, the more response you're going to have from the client and the better results are going to be. Revive NX contains 150 billion exosomes per vial, which I think is definitely the largest number I've seen available. So it will work very, very well. It also contains this nitrogen 1, which is patented technology. And this is a molecule that strengthens the dermal epidermal junction. So it basically sticks the skin layers together. And finally, it's about where they've been derived from. And I think we have a slide on that later. So I'll hold off for now talking about where we derive them from. But I'm sure you'll all have the questions of, what do I do? Is it plant-based? Should I use human-derived? Should I use animal? Should I use bacterial? And I'll tell you about that shortly because it is really interesting. So, looking at the safety and the ethics of exosomes, because if you've done any research on exosomes, you will see that there are some questions around certain exosomes, and I want to put some of your minds at rest about this. Revive NX exosomes are bacterially derived. So if you've looked at exosomes in the past and you've looked at anything around human derived, you'll have seen that there are some ethical and some safety complications around those. Ethically, we are not legally allowed to use human-derived exosomes in the UK. Safety-wise, human-derived are more likely to have you're more likely to develop reactions from human-derived, and we don't want that. The other thing is that the Revive NX exosome, because of this, have been given the regulatory approval within the UK for use. And because they're not human derived, it does avoid that risk of immunogenicity and immune reactions from your clients. So this is the important slide I want us to look at. What exosomes are what? Why do we want exosomes? If we want exosomes, which ones do we want? Because you'll go around a trade show. I went around a conference in Turkey, by the way. They are massively into mesotherapy, exosome treatments, biostimulators. It's that's the big thing over there now. And every other stand was exosomes. And it's like, what makes it good? And if you go to any of the shows in the UK, you'll see the exact same thing. There are so many people saying that their exosome's best. You already know you want to look at the concentration, how many exosomes are in there. If it's got nitrogen 1, which it won't, unless it's Revive NX, but any additional things are worth looking at. But really, really importantly, is the type of exosome that's there. Where's it come from? The main ones in the country, or the main ones available in the world, should we say then, are human derived, which we've already said in the UK have ethical, legal, and safety implications, and we can't use them. We have animal derived, which is bovine, usually. So bovine milk, often, or bovine colostrum. We have plant-based, and then we have Revive NX, which is bacterial based, and that stands apart from most of them. Now, the example I always give, because a friend of mine who's been in the industry for years told me this, and I think it's a really good one, is you've got to think about exosomes as a message. It's relaying a message. So it needs to be speaking the same language, which is why human derived, if we're able to use them, would make a lot of sense, because that would be the same as me talking to somebody else who speaks English perfectly, and we'd have the same language, use the same words, we'd understand each other 100%. We can't use them, so we have to look at an alternative. If we look at bovine, genetically, we're all, us and cows are mammals. We separated how many millions of years ago, but we share similar DNA, and therefore, I don't know the exact number, but we probably share about 98%. We share 99% with the chimpanzee, so it's probably about that. So actually, a lot of the messages are going to be the same. And so we look at that as if I was talking to somebody who was speaking French. I did French at GCSE. I can't speak French, but I could probably follow a bit of the conversation if they were quite slow and I pick up enough to understand what they were saying. Right? That's like bovine exosomes. They're going to speak to your cells and you'll understand most of it. Think about plant exosomes. How long ago did we split off from plants in the evolutionary tree? Millions and millions and millions of years ago. Our DNA is not very similar to a plant anymore at all. And therefore, trying to get a plant messenger to tell our cells what to do isn't really going to work, is it? And again, the example I give there is that I haven't got a clue about, for example, Chinese language. Me trying to have a conversation with somebody in Chinese or somebody who'd never spoken a word of it in my life, I wouldn't have a clue what was going on. And that's similar with plant-based. So where does bacterial come in? The bacterial derived exosomes here, the lactobacilli are a commensal on our body. So they live on our body. I don't know if you all know this, but we have many more cells in and on our body that aren't our own than are our own because we have so many bacteria, viruses, etc., that live on our body. These are a commensal, which means they live there, they don't cause any illness, and we live in harmony. Most of our serotonin that's produced in our body, the happy hormone produced in our body, isn't produced by us in our brain. 90% of it is produced by bacteria in our gut. So these commensal bacteria make a big impact on our lives. And for millions of years, this has been the same relationship. We've got this symbiotic relationship. We communicate with them, they communicate with us, they make things for us, we make things for them, and it works in harmony. So this is why lactobacilli bacterial exosomes make sense because they're not human derived, because they're bacterial derived, but it's a bacteria that for millions of years has been communicating with us and we've been communicating back. So we understand each other's language. And that's why bacterial makes sense. Right. So,
Sorry, Doctor,
yeah.
can I just ask you, we just had a question. Do you have any information on salmon-derived exosomes at all? Have you
Don't
worked?
have information other than I'd probably personally put that between the bovine and the plant because just think about it from a evolution point of view. When did we split off from plants? When did we split off from fish? When did we split off from cows? Yeah. Right.
Okay, so it's the DNA compatibility with our skin. It's not there, just like it wouldn't be with a plant. We're very far away from speaking the same language, so the compatibility is not there. Therefore, the result won't be there.
No.
Right?
No.
We don't get the optimum result if we're using which is very different from how polynucleotides work, which we're going on to. We're talking about the cell-to-cell communication,
Yeah.
two cells, and the DNA messaging. And so what we're understanding is that obviously we can't use human-derived, and the bacterially derived exosome is the most compatible for best results in cosmetic aesthetics. Yeah,
Yeah.
that be okay?
Yeah. It's different when it comes to the polynucleotides that we see because essentially your exosomes are giving the message and your polynucleotides are providing the sort of the,
Stimulation
the
tissue.
bits that you need to
Yeah.
do it. So you can use that. We're not relying on polynucleotides to necessarily provide a message.
Oh, the DNA code, like the DNA mess.
Yeah,
We're not providing that cell to cell message. It's a completely different modality. So, therefore, fish-based derived exosomes would not be compatible for the best results as with plant. Okay,
yeah,
that's brilliant. Okay, any more questions? If you don't mind, I'm going to just pop them into you whilst we're on the subject, but we'll
yeah.
try and keep it on the question and answer. Thank you.
That's all right. So yeah, any questions, by the way, yeah, please guys, pop them in the chat and we'll get to them at the end. Um, so be aware when you're out there shopping for exosomes, because again, I'm not going to force Revive and X down your throat. They are my favorite, but you guys have a shop around once you've learned from today and see what works for you. Um, you've got to ask yourself where they're from, how concentrated they are, and do they have the nitrogen one in there? But have a look around. The other thing to be aware of is companies falsely claiming that their products are plant-based or animal-based or something like that. They can all, when I first started speaking to the guys at Revive, I asked them to send me the paperwork across, and I got the paperwork that told me everything I need to know from a science background point of view. Um, Companies should be able to provide that. If they can't provide that, if they're a bit sketchy about it, or they're like, oh yeah, it's a plant, or I'm not sure, it's probably because they're hiding something. And some people are hiding and using human-derived exosomes disguised as plant or something else. While that might seem like a good option, one, it puts your patients at risk. You don't want to have to be dealing with an emergency anaphylactic reaction as a result of it. And two, it would absolutely void your insurance. So please avoid that. And if they can't give you the evidence of where it's derived from, I'd stay well clear of it. With Revive NX, it's bacterially derived, as we've discussed. There's no human DNA or any immune risks with that. There's no ethical concerns because it's bacterially derived. It's fully insured and widely used across Europe. And the FDA approval is pending for microneedling in the USA. When we any human-derived exosomes not available in the UK are sourced from umbilical cord, adipose tissue, stem cells, etc., and do have risks associated with them, so stay well away. Right. I think we've covered exosomes. I want to briefly look at polynucleotides. This isn't necessarily the focus of today's talk, but we are looking at how we combine polynucleotides and exosomes. And many of you out there are probably already using exosomes. You're probably already using polynucleotides, and you don't know how to get the best of both out of them. So, polynucleotides are purified DNA fragments that act like a blueprint for skin healing. They attract fibroblasts to the area. They stimulate collagen and they enhance hydration in the skin. And they're especially useful for patients who have crepey skin, poor skin barrier, pigmentation. I've had some great results. And for anybody with sort of mature skin, sun damaged skin, etc. Um, Clients love them, practitioners love them. With any regenerative treatment, you always have to kind of warn the patients because if they're used to having fillers where they get an instant result, that isn't going to be the case. It does take some time, but the result that they get at the end is way worth it. So, how do we use polynucleotides? Many of you out there probably use injection, to use a needle technique. Um, and this can be really good, but as you may have found, it can be uncomfortable. If it's not uncomfortable when you're doing polynucleotides with a needle, if it doesn't sting, your polynucleotides aren't very good. Unfortunately, the high strength polynucleotides are the ones that sting, and that's what you want to get your results. Um, but the other thing that happens that you can avoid is bruising, particularly under the eye area. Polynucleotides are fantastic under the eye, for people who have crepiness, dark circles, sunken eyes. They don't want fillers, or you don't think they're a good candidate for fillers, particularly if they've got puffiness under the eyes, you want to stay well away from it. Then polynucleotides are brilliant. The downside is that they bruise because you've got tiny little blood capillaries under there that are one cell thick. They have no muscle in them. They can't constrict when they get damaged. Therefore, when they bleed, they keep bleeding, and you can end up with a lot of bruising. And if someone's got to come back for 3 treatments of polynucleotides within the first 3 months, so they're going to spend most of that 3 months either having a bruise, recovering for a bruise, or waiting a week until they come back for more bruises, they're very unlikely to finish that course of treatment. If you're doing accordion lines, you can end up with a lot of injections along there and it can be uncomfortable. So instead, we might want to use a cannula. Cannula is great for this. The one thing I would say with a cannula on polynucleotides is if you're using a cannula for fillers, you're going quite deep. For polynucleotides, think about where you want them to act. You need them to be at the surface layer of the skin. So you're going very, very superficial with your cannula. So if you haven't done it already, there are ways that we can train you in that. We'll talk about my training courses later in case it's something you haven't done yet and you want to come along. But you can use needle or cannula for these treatments. So just like we looked at what makes a good exosome, what makes a good polynucleotide? Firstly, the purity of the product is really, really important. The concentration of the product is really, really important. There are products out there that I've seen that have 2.5 milligrams of polynucleotides in them, and there are products out there that I've seen that have got 40 and 50 milligrams of polynucleotide in them. So make clever choices. If you want results, you need higher concentration. If you get something that's got 2.5 milligrams in there, it's not going to do a great deal. One product I use, which I won't name, but one product I use, their eye product is 15 milligrams. Their face product is 40 milligrams of polynucleotide. If I do their eyes, I start with the first 15 milligrams. If they respond well to that and don't get a lot of puffiness afterwards, I'll put them on the 40 milligram next time. Because if you think if I do three treatments, 115, 140, 140, compared to somebody doing three treatments of a product that has 2.5 milligrams in it, where they get 7.5 in total, they're not even getting half of the dose that they would get from the first dose of the weakest version of the better product. So look at the concentration, that's so important. You want minimal additives, um, and you want to make sure that you've got a reputable product that's come from a reputable pharmacy that's had all the safety um, um, research done on it so that you know you're getting good results for your clients. You don't want to inject something you don't know much about. So now, importantly, exosomes and polynucleotides, why do we combine them? It's going to be more expensive for your client. So why do we want to combine them? What's the benefit there? How do we sell this to our clients? When we were talking before about fish exosomes, and would that be right? Whereas polynucleotides are often salmon or trout based, and that's fine, but it wouldn't be fine in exosomes. Why is that? Exosomes signal the skin to act. So exosomes are the message to tell the skin to do something. That's why that needs to be clear. That's why you want the bacterial derived or something that what the body understands. Polynucleotides provide the tools for repair. So when you use them together, they deliver superior results because you're given the instructions to work and you're given the materials to do it. It's like telling a builder to build a wall, but you haven't provided any cement or bricks or a mixer or anything. Whereas if you go, builder, I want you to build a wall, that's the exosome. You've told him clearly in the language that he understands, build a wall. And then you've also done, here's all your stuff that you need to make that wall happen. It's going to work much better, isn't it? So this is great, particularly in complex or sort of resistant treatments. You've been doing some microneedling with exosomes for a while. You've done three treatments. The patient's still not hugely happy. And actually, when you look back, you think, you know what? They had such sun damaged skin when they first came in. They've been using sunbeds for 30 years. They're in the 60s. What was I really expecting the exosomes alone to do? I probably need to think about adding in some polynucleotides to really turbocharge that. So, how do we integrate this into a protocol that can work in our clinic? There's a few different ways. I think when anybody comes in, let's take it as a client journey, okay? They come in, they've got volume loss in their cheeks, they're in the 60s. Um, they come in, they say, since I went through menopause, I've just aged. I've got fine lines and crepiness. I've got volume loss here. I've got nasolabial folds. I've got marionette lines. You think, where do I start? The new way of doing this, the way that we should all be doing this, forget about volume straight away. Don't jump straight in with your fillers. Sort out the canvas first. Treat the skin with your exosomes and polynucleotides. That's going to stimulate collagen. That's going to improve the structure. Then think about adding filler if you need to. So for this client, if it was me, I would start with exosomes and microneedling because that can give you a whole face, under eye, and neck treatment. I would treat them with exosomes. I would then bring them back in around 3 weeks later for a course of polynucleotides to start. You can do these at the same time, but think about the way your skin regenerates. It takes 6 weeks for the skin to grow from the basement membrane up to the surface that you can see. As we age, that process slows down, so it can be more like 8 weeks or more. If I give you everything at week one and then wait 6 weeks for the next treatment, I feel like that's a missed opportunity. So if a client wants both and can only come to you once every 6 weeks, fine. You know what? Do it in one sitting. But if you can get them to come back twice throughout this or multiple times throughout this treatment, it's going to be better for you. So exosomes on week one, bring them back at the midpoint around week three, and think about some polynucleotides for particularly stubborn areas. So if they came in and they've got a lot of accordion lines down here, target it with polynucleotides. 2 or 3 weeks later, repeat the exosome treatment. 2 to 3 weeks later, repeat the polynucleotide treatment. 2 to 3 weeks later, exosomes. And you want to do 3 of each. So 3 lots of exosomes, 3 lots of polynucleotides over a 18-week period. You can do 6, 12, 18 weeks. This is going to give them the maximum results. And even after this is done, it's still going to take around 12 weeks for them to see the final finished results. They will get results after 6. But if you say to them, we do this course of treatment for you, give it another 3 months after that and see how good you look. And then they just come back for simple maintenance treatments. So once every 4 to 6 months, they may come back in for the polynucleotides and their exosome for microneedling. It really depends on the patient. Some of them I've had where they'll come in every 3 months, some every 6 months, and some it's a monthly series. So it just depends on them. But your initial protocol is this: exosomes, 3 weeks later, polys, 3 weeks later, exosomes, 3 weeks later, polys, 3 weeks exosome, 3 weeks polys. And you're going to get some fantastic results. Right, I think this is you now, Nita, isn't it?
Right. Yeah. So what we're talking about is business integration. Right. So we know about our modalities and the science behind it, but how do we actually put it into place? Because there's one thing knowing about the product. It's really good. You must educate yourself on the science, because if you don't know what you're doing, why you're doing, how can you upsell it to your clients? But then how do you integrate this into your clinics and businesses, academies? The great thing is that we work with the Child Bio Group, which is South Korea's oldest and largest pharmaceutical manufacturer, medical research company, and it provides raw material, pharmaceutical materials globally. So we hold the patent to nitrogen 1, which only we have in our exosomes. This is not available to any other exosomes globally either. So this makes our product far superior. The business integration. So our ethos is always sales through education, which we like to transfer to our practitioners and our academies and our doctors. We educate our client. They know why they're coming in, what they're coming in for, what they're going to achieve. Now, 2026, we're entering the year of regenerative aesthetics. So this is great. You're here, you're educating yourself well in advance. We know that this year was all about undetectable injectables, but 2026, you're going to hear a lot more about exosomes, cross-protocol applications to your clients, because we're living now in a culture where a lot of people want results fast, right? However, fast results need downtime. So you can't come in at lunchtime, have an exosome polynucleotide combined protocol and expect no downtime and look like a Hollywood celebrity the next day. So what I would recommend is that everything you learn, everything that you need, everything that you think that your client would need, the good, the bad, and the ugly, but obviously the benefits far outweigh some of the downtime, like the downtime that we may have, because we're all different. Put it into your informed consents. Make sure you're protecting yourself. We're dealing with powerhouse treatments. So powerhouse treatments that give powerhouse results that often need some form of downtime. So that's really important, to make sure that you're communicating that with your clients and they know that they're entering these treatments fully informed. So going forward, the other thing that I like to concentrate on is the relationship that you're building with your client. So it's just not just about this is why fill and freeze, moving away from it's quite great, because you're going into regenerative aesthetics, you're going to see your client more often. You're building a long-term relationship, creating a roadmap and a treatment program for them. You're keeping your client close, so you're having the repeat client coming in. And we all know that your most familiar clients are the easiest ones to manage. They stick to the protocol, they commit, and you're also seeing the results coming through. All of this also, as Dr. Lord has explained, the skin needs time. We need time to get results. So these are not instant results like a filler or Botox you're going to get in 2 weeks. The skin needs time to regenerate and get the results. So that's one thing you've got to understand. Now, the great thing is about the access that you have to our exosomes is we're not the cheapest, but we're not the most expensive, and our product range is generous. So you've got to get a lot of bang for your buck, but you get the result. You're going to have very happy clients, the clients that know what they're entering, clients that know what you're doing for them. You are going to get the results. So what we're looking at is increasing your quality and your results, decreasing your expenditure, and maximizing that profit margin for you and your clinic. Because let's face it, we do want to make money, but you're only going to make money if you implement all these things that we've taught you today in the correct way. Okay, so please play if you can, if there's any more bullet points on this slide. Am I still sharing, or is that you sharing, Keiron? Yeah, so I've just gone through that. So you've got superior access. So we know that Korean technology at the moment is taking over aesthetics. So we are working with the one of the most established pharmaceutical companies called Bio Group of South Korea that provides raw materials to the world, and they understand the compliance levels of each country. So as you can see, we want to increase the client retention. We want to see them more often, and we covered earlier on that fillers still have their place for profound volume loss. They still have their place. They still work brilliantly. We've also got a brilliant range of dermal fillers. We also have another modality for collagen stimulators that we can cover on a different webinar with Dr. Lord. And we also have so that will be the 5-step regenerative process. So we would repair the canvas, rebuild the skin, go in with the fillers, hydrate the skin with our hydro boosters, and finish off with home care. So that's the regenerative roadmap that we have to keep our clients knowing where they're going, us keeping our clients close with us, and there's a treatment roadmap. So that's what we like to help you to bring into your clinics, your academies, how to teach it, how to give it to your end user. Okay, so if I can move on to the next slide, please. So we talk about the support. Right, so everyone that joins the webinar and anyone that purchases our product. Now, unfortunately, we can only supply to UK and Ireland. We do have our partners in Europe. So if you're in Europe and you've joined us today, we can direct you that way, but we can't supply outside of UK and Ireland. But anyone that does the training with us, so if you can't purchase, you can still purchase the training. That's totally fine. You will get support, because if you succeed, we succeed, myself and Dr. Lord and our brand. We want to help you to succeed. So you can do our training with us when you purchase a box of Revive and X exosomes. This, once you've passed it, will give you CPD accreditation, online accreditation for our exosomes. You will also get an authorized practitioner emblem that you can use throughout your social media, your websites, and can link back to us. And then when you've got your before and after results, we will share it. So tag us on all your work. And this is just for anyone that's completing our training and buys our products, so that you can say that you're authorized, and our training levels are high up. So we've got our regular webinars with Dr. Lord, and as the aesthetics industry is evolving, he is imparting all this valuable information. So it's CPD accredited, and you'll get your unique practitioner ID with it as well, with your certificate. So that's the support that we give for all our practitioners in our academies. We've got a separate program for you as well, and you've got an opportunity to become an ambassador for us as well. So that's our support program. Next slide, please. So please, if you can follow us, follow Dr. Lord. He's always doing exciting webinars. These are our accounts. Please, if you're doing before and after pictures and you've got great results, tag our head office as well. Tag all of us so we can reshare your brilliant results and support each other. So our cross protocol, why we've done this is that we want you to be able to maximize profits as well. So what we're saying is that we also have the time constrained client that may not be able to come in for a roadmap every 3 weeks. But is willing to take some form of downtime and have exosomes and polynucleotides in one session, there will be a bigger downtime, and you've got to make sure that your client is informed for that. But that is a protocol that we are using throughout the EU and is one that is an option to you. But obviously, as Dr. Lord has explained, it's always good to give the skin a rest to get the best result. But you've got that option where if you're a time constrained client that has got that is willing to take some downtime, you can do it in one session, or has got the budget, or you can make a package for them. So that's a way that you can implement this in your clinic. And then the next slide, please. Yeah, no, the one after. Right, so I'm going to go through a few things with you. Please screenshot this if you can. Right, so Dr. Lord's got an opportunity for in-person training. Now, this is in Devon, in Exeter. It is fantastic. We did a roadshow with Dr. Lord only about a month ago, 5-6 weeks ago, and the knowledge that you're going to get, if you think that you've got a lot in this webinar, it's going to be, you get access to Dr. Lord in person. You'll be able to go through polynucleotides, exosomes, cross protocols, and you can pick his brains on everything else. Also, sorry, our next webinar that we're doing is on our brand new product, which is Revive NX Hair and Scalp Serum. That's not going to be until February because both Dr. Lord and I have a very heavy schedule. So that's going to be in February. We'll send out the date to everybody as soon as we can. And that is on our new product, which is Exosomes with PDRN for the scalp. Now, this is amazing to prepare a canvas for hair transplant, post hair transplant, a step into hair rejuvenation, hair and scalp rejuvenation if you're finding thinning with menopause, menopause, and all sorts of transitions that we have in our lives. So that will be our next one. We will have further webinars coming up. So any subjects that you'd like to cover, please drop it into the chat and we will be able to discuss it between us and see what webinars we can provide for you. And great news I know that we've got some European partners and doctors that have joined tonight and will be rewatching this. Dr. Lord will be with us at IMCAS. So he will be with the Char Bio Group, Char Meditech, and representing Revive NX Limited at IMCAS in Paris at the end of January. And for those of doctors that are joining the gala, we've got 6 doctors, and Dr. Lord's one of us that's going to go to the gala for Revive Next, yay! So he'll be representing UK and Ireland, which we're very proud of. So you have the opportunity to meeting us in IMCAS if you're attending. And yeah, I think I've covered everything for that. For more business integration, we've got all the protocols and more ideas on to help you do some of the basics that you may not have thought of on the portal when you do the training as well. So thank you for coming on board. We will be having this up on the portal and a private link for everybody to rewatch later. And I want to thank you, Dr. Lord, for doing our webinar again, and we will join you again in the new year. Do you have any words?
Just thank you to everyone for coming along. Thank you for spending your evening with us while we talk to you about our exosomes and how you can integrate them into your clinic. I do think, well, this is definitely the way regenerative aesthetics is going in the new year, and I think there'll be a lot of demand for this. So absolutely, if you aren't already, integrate exosomes into your clinic. If you want to know more about polynucleotides, exosomes, then very happy to answer any questions. You know, drop me a message on Instagram. And the online, the in-person training down in Devon, we are doing it around the country though. Devon's just the first one that's available in January. That is looking at different protocols for different skin types. So there are various polynucleotides out there. Some have HA in them, some don't. Some are higher concentration, some are lower. And we can look at that in person. We can teach you skills such as needles versus cannula, and you'll be able to take that straight back to your clinic. It's all CPD accredited. So yeah, if you want to know more about that, get in touch and ask me any questions. And with regard to the next webinar, all around hair restoration, hair regeneration, that is a massive area where you can treat people anyway, but particularly tapping into that group of people who are flying over to Turkey often to get a hair transplant. I was in Turkey last week and I was chatting to one of the clinic owners out there, a big 81 room clinic in Istanbul, and they don't have any partners in the UK that provide this service. So think about how hair loss happens. The scalp isn't working as well as it should because it's not very healthy. You optimize that scalp pre-hair transplant. They go to Turkey, have the hair transplant. They come back to you for ongoing maintenance. If you could link up with clinics like that, that's a really good option for you from a business perspective. So come along to that webinar in February to find out more about that product.
I just got one quick question before we close the webinar. Is it correct that hybrid PN-HA product is more superior than PN on its own, polynucleotides on its own, even if hybrid product has less PN than PN-only products? What is your opinion?
No, it depends on what you're trying to treat. You know what? I think HA and PN product together works really well for things like the lips. So if you've ever dissolved lip filler and then people come back and they've got the only way I can explain it is baggy. Like they've had so much filler, you dissolve them, they just become really dry and crepey. And then I think HA, PN, a couple of treatments of that pre-replacing lip filler is really, really good. Other than that, no, it's kind of you got to make the choice. If your patient's got a combination of crepey skin and dry skin, you might use HA and PN. If it's mostly crepey skin, then stick to a high concentration polynucleotide.
I agree with you. I think it's great to kind of separate them and making sure that each treatment is, so you're doing a high quality HA, doing a high quality PN, you're giving a gap for the skin to repair.
Yeah.
Guess what? You can actually microneedle exosomes to the lips as well for the repair as well. I think we discussed it on last time, but we didn't cover it today. But that just reminded me.
Yeah. If it was me, I'd rather use high dose, high quality polynucleotides and then throw in a skin booster on top than use a weaker polynucleotide just because it's got HA in there.
Yeah. So we're not dropping the quality of any of these products just to combine them in that once. We're just doing the high quality by separating them. That's great feedback. Okay, so just quickly check. There's no more questions. So thank you so much, everyone, for joining us, and we'll catch you on the next webinar. Watch out for our emails. We'll let you know when that's on. Thanks.
Thank you, everyone. Bye bye.